Oh, Mercedes.
Oh, they also make e-bike. So I just bought it at Tor.
You bought a Mercedes?
No, I got it at Tor e-bike.
Oh, nice.
And then I found that Mercedes also had e-bike.
Yeah, Mercedes doesn't make bikes, so I wouldn't.
Okay. Yeah.
That power, I think, is like if you want to spend like not too much money, you want a solid like electric bike. Like,
Yeah, I rode it yesterday after assembling it. Really nice.
is it fun? It's nice.
It's fun. Yeah.
Oh, nice. That's good. Biking's a lot of fun, especially. Can you bike here? Can you do that?
That power I can. Not throughout, but I will take this metro. So I will take the metro line.
My first year at USC, I used an electric skateboard to get to campus every single day from downtown. And the day before my screening exam, I got hit by a car. Biking's a lot of fun. I prefer manual, but
No, I have a road bike, but with the hills, like
yeah.
Okay, then we can get started. Okay, so thanks, Jessica, for coming. We have Jessica Lee, who's an associate professor in the physics department at UCLA today. Thank you.
For the kind invitation, it's my pleasure to present here again after 7 years. Maybe that my last visit was like 2016 or 17. So yeah, I'm very happy to be here to present our recent work. So this is a method computer, and it's a joint work with my two former PhD students. So our goal is actually to do some feature selection or feature screening, whatever you call it, without using keywords. So to our applications in genomics and biology, this is a very commonly used task, and many people thought this problem is actually done, but we do see a lot of glucose in the current practice. So that was the major motivation of our work, and I'll talk about that. Move my mouse back. Okay, so here the background is that we're working with high throughput biological data. By high throughput, we mean that we measure high-dimensional features simultaneously, usually at least 10,000. And we have a very small sample size because it's very expensive to generate those data. So usually the sample size is often no greater than 3. So the most common analysis is to compare two conditions, which means that we have 3 replicates under each condition, and we want to compare the two conditions as one called experimental, usually the condition of our interest, for example, with some mutant or with some experimental treatment, and the other is the background serving as the negative control. So in this typical setting, the goal is to identify some interesting features to biologists. What do we mean by interesting? So as you can see here, the problem is we're doing marginal feature screening, one feature at a time. And by interesting, we mean the feature as called interest enriched or differential. So for enriched features, they are defined to each satisfy that it's expected experimental observation, that is, its expected observation under the experimental condition is greater than its expected background observation. So I'm going to talk about 2 applications under this setting. The first one is called p-calling from ChIP-seq data. So this is what the data looks like. Usually we only have 1 track per condition or sample of size 1 per condition. So here we divide the genome into those bins. And for each bin, we're going to decide whether it's count. So this is like a peak. We can summarize this into count. Whether its count is larger, have a larger expected value under the experimental condition than the background condition. So here the features are genomic regions or bits. The second example is to do this so-called peptide identification from mass spectrometry data. So the data looks like this. So we have each spectrum as this. It's a called mass spectrum. And what we do is that we want to find its corresponding peptide sequence in the database. So this is a real database. What the algorithm does is to compare the spectrum with the database using their own way and output a pair. So each spectrum is going to be matched to the best sequence and with a score. So this is called a matching score or alignment score. But how do we know the score is high enough? We have a negative control. The negative control is based on a decoy protein database in which we actually reverse each peptide sequence in the true database with the belief that those decoy sequences are false. So therefore, we input the same spectrum into the decoy database, and we also have the pairs with scores. So you can see this red spectrum is going to be matched to a decoy sequence with a score. Then the idea is to compare the scores for each spectrum between the target pair and the decoy pair. See if the target pair has a much higher score than the decoy pair. If it does, we will report the sequence as a matched peptide. So that's the rough idea. And as you can see, this is one directional comparison or one-sided comparison. Okay,
Got a
that's,
quick question here.
yeah.
Sorry, so are you comparing those two red pairs because they correspond before you made it, or are you comparing them because they were the two with the highest scores?
No, I compared like the red pair, the green pair, the yellow pair, because each pair is from one spectrum. So it's just the same spectrum matched to a target sequence, matched to a decoy sequence, and we want to see if the target score is much better than the decoy score. If it does, then we think the target match is trustworthy. That's the idea.
Is that like the knockoff kind of thing, or
Is that what?
is that like the knockoff?
No, we're not constructing anything. But the idea is like this can be regarded as a knockoff, a fake thing.
So the decoys are truth or?
Are false. So we actually manually constructed the decoy database by reversing each sequence in the target database with the belief that these reverse sequences are not true. They're not in biology.
Okay.
Yeah, thank you for the question. Yeah, so this is also an enrichment analysis. And for differential analysis, we expect to find the features with different expectations under the two conditions. So two typical analyses are called differential express genes or differential interaction regions. So for differential express genes here in this example, under each condition, we have 3 replicates and each column is 1 gene. So usually what biologists does is that they look at this, 3 points, sort of like rough box plot to see whether the gene has higher expectation under one condition than the other, right? So in this case, the features are genes. So in this case, the left one will be called as a differential express gene. The right one may not be called a differential express gene. Okay, finally, this is what we call the 3D genome data. So this is basically like a half of a matrix. Okay, we only look at one triangle. And here, what it means is that we can consider these as rows, these as columns, and they are symmetric. So basically, for each row and each column corresponds to 2 genomic regions or bins. So we look at the entry as their so-called interacting intensity. This is measured by experiments, meaning that the higher the intensity, the more likely the 2 regions are contacting each other in many cells. Okay, so usually this experiment is very expensive, so we can only do 1 experiment or 2 experiments for each condition. So here we have 2 replicates, and what we do is that we want to find the blocks where we have very different intensities between the 2 conditions, and those are called differentially interacting regions. So as you can see here, each entry is a feature. And the features are called region pairs because it's a pair of this region and the other region. So this is differential analysis. So then what is the criteria people commonly use for setting the cutoff? It is the frequentist FDR. So this is the most dominant criterion people use in this type of analysis. And as many of you know, it's defined as expected ratio of the number of false discoveries over the number of discoveries. Although we have under the Bayesian paradigm some analogs like the Bayesian FDR, local FDR, or more recent, there's a local false binary. But these are still not so commonly used compared to the frequentist FDR. So that's my focus today. So what is the current practice for controlling the FDR? P-value based method is the dominant one because of this famous Benjamini-Hochberg procedure, shown as BH procedure, one of the most well-cited statistics paper, right? So here, just a quick review. What it does is that it will sort the p-values from the smallest to the largest. Let's say we have D features, so we have D p-values. And for the ith order p-value, we will compare to this number. As you can see, the larger the d, the smaller this number, right? So it means that if we want to make discoveries by finding the p-values such that they are under this threshold, we really need the p-values to have a fine resolution, very fine p-values. Otherwise, we may not make any discoveries. And we have the Story's Q-value procedure, which is also popular, and it also requires high resolution p-values. And these two p-value based procedures can achieve exact control on the frequentist FDR. For approximate FDR control, there are the local FDR based method. So this is proposed by Efron. So what it does is that we'll try to calculate a local FDR value per feature, and then threshold the local FDR value directly to the FDR threshold Q to approximately control the FDR. Although this is not exact control, but some people still use it. Okay, so here our motivation is this requirement for valid high resolution p-values in current practices. As you can see, if we have a very small sample size to have high resolution p-values, we must rely on distribution assumptions that are parametric tests. If we don't want to do parametric tests, want to do non-parametric tests, then we need a large sample size, right? So we can do permutation tests that can give us some valid p-values with reasonable resolution. But that we can see, even with a large sample size, the number of permutation will determine the resolution, and that must be large and can be computationally intensive. So I will talk about the two approaches for calculating p-values in the current practice, paired and pooled. Okay, so what does paired mean? It's basically what we call two sample tests in statistics. We examine one feature at a time to compute a p-value for that feature. Okay, so this is used in the p-calling application, differential express gene application, and the differentially interacting region application. So three of the applications I mentioned. But the two common issues we saw in current practice is the model, or I would say test misformulation. In other words, this should be a two sample test, we know, but people actually did one sample test, as we'll see. Next slide. And also another reason is the model misspecification. Okay, so back to this p-calling analysis, we want to compare these two counts, okay? And so this is a very famous software published in 2008. It's called Max for doing this test. So far it has been cited for more than 10,000 times. So it's a very popular software. And actually I happened to read this paper when I was a PhD student, and I had a confusion here. So this is what it did in a very simple summary. So I have to acknowledge that the algorithm is more complicated than this, but this is the essence of the statistical part. So for a region, okay, they consider two random variables. One is its background count, one is its experimental count. And here I'll have to say its sample size is 1 versus 1. We only have one replicate per condition. Okay, and for the data, the observed value denoted by small x, small y. So the way it calculates the p-value is this probability. The probability that this big Y is greater or equal than small y, with the big Y assumed to follow Poisson distribution with mean small x. Is this correct? Actually, I had this question when I read the paper at the first time. I don't think it's correct because of the following reason. If we really think about the logic behind this p-value, what it actually does is try to assume that big Y follows Poisson with parameter lambda and test these two hypotheses, right? The null is lambda equals small x. The alternative is lambda greater than x. So in this formulation, you can see that the small x is treated as a fixed parameter and its randomness is actually ignored. So this is actually a one sample test formulation, but we know it should be considered as a two sample test. But back then, I didn't have a good solution myself to fix this because I thought, oh, the sample size is 1 versus 1. What can we do, right? In our textbook, we never taught examples like this. Maybe there's no good answer. So that's one motivation of this work I'm going to present next. And second motivation is this model misspecification. This is another popular method for a different task, for this differential express gene task. This method called DE-seq2 has been cited for more than 30,000 times, okay, for doing this analysis. What it actually does is that it will assume a gene follows a negative binomial distribution under each condition. And what it tries to test is whether the two negative binomial distributions share the same thing. Okay, but recently we found that applying this method DE-seq2 to a data set about cancer immunotherapy, we found a very surprising phenomenon. This is DE-seq2. The red dots denotes the number of differentially expressed genes, shown as DEGs, from the real data. And we did a lot of permutations because here the sample size is hundreds, so we could afford to do permutations. On each permuted data, we just scrambled two conditions, so the samples are actually not distinguishable anymore. And on the permuted data, and this bar plot with the error bars indicate the number of DEGs identified from permuted data. So very surprisingly, we found that on many permuted data sets, we could identify, so the DE-seq2 method can identify more DEGs than it does on the real data. So this is definitely a very strong indication of a likely inflated FDR, false discovery. So we actually looked into this, and we found the reason is that on this data set where the observations are from individuals, like persons, instead of experimental replicates in the lab, the negative binomial assumption or the distribution does not fit the data very well. Because we have hundreds of observations, we can check the goodness of fit, and we found it didn't fit well. The same issue applies to another popular method, HR, so they are competitors. They both assume negative binomial distributions. We found that, oh yeah, they both have the same inflated issue, that inflated FDR issue. So with this, we can see that commonly used parametric model may not work well on any data set, but the problem is that most users in the field don't have the expertise to check the model assumptions. They just go for the most popular model method and use it. So this is really surprising. And in our bioRxiv paper, we also showed that the falsely discovered, differentially expressed genes, many of which are functionally related to immune response. So in this case, the false discoveries may look like true, and the biologists will tend to report that finding by saying, oh yeah, the cancer immunotherapy treatment is effective. It changes the immune response gene expression, but that's not the case. So this is the second motivation.
Sorry, just one.
Yeah.
Isn't it true that method is often applied to data sets with very small sample sizes?
Exactly.
So even if people wanted to check the model assumptions, they would have no way of doing
Exactly. Yeah. Usually when the replicate number is 3, how can you check the goodness of fit, right? You have to just believe the model. Yeah. Okay. So that's a very good comment. And the second approach is not pair but pool. So this is a cartoon illustration of what it does. So this is applied to that mass spectrometry peptide identification task. So from the background data, we get an average for each feature, and then we pull those observations, the averages, into an empirical distribution, okay? And then for each feature measured under experimental condition, we get their averages, and for each xj bar, we put it into the null distribution and find the right tail probability as the p-value. So this is what people do in this view. And as you can see, the assumption here is that the features should follow a homogeneous background, which means that they should be IID under the background. But we know that this assumption may not always hold. Okay, so we see enough problems with p-value uses or calculations. Here's our proposal. We want to propose a method that does not require valid high-resolution p-values. It doesn't need to assume parametric distributions for data, and doesn't require a large sample size. So to achieve this, we were very fortunate to find this very useful tool developed by Barbara Candace in their knockout paper for achieving FDR control without requiring high-resolution p-values. So motivated by that, we come up with the idea for the above analysis invention to just use 2 components without p-values to directly achieve FDR control. The first component is what we call the contrast score, 1 per feature. So we use this name because we think it's intuitive and makes sense to biologists. And the second is to set a cutoff on the contrast scores using the BC procedure, or we also consider this GZ procedure by Guinness and Zhou from their multiple knockoff paper. Okay, so why do we choose this main paper? So just two reasons. One is that we think we're trying to clip up a small number of features, and second is because we're based in LA, so of the MBA team. So we hope this method to be robust to data distributions, sample size, and also the existence of outliers. So the goal is that if we want to choose a robust statistic for the contrast score definition, then we can make it robust to outliers. Okay, so here are some notations to formalize the discussion. For feature J, we consider its M observations under experimental conditions as this M-dimensional vector. Its N observations under the background condition as this N-dimensional vector. Okay, and because of the motivations in my examples, it's reasonable to assume all the values here are non-negative and they are IID under each condition with the same mean. So for feature J, its experimental mean is denoted by mu xj, background mean as mu yj. So following our previous definition, enrichment analysis to find features with mu xj greater than mu yj. Differential analysis to find features with mu xj not equal to mu ij. Okay, so what we are assuming so that the BC procedure can be applied is independence. So what we mean by that? So conditioning on all features expectations, we assume that for feature J's M and N observations, they are mutually independent. So we are assuming basically the measurement randomness are independent. To us, this is a reasonable assumption for experiments. And the second thing is that because of the second assumption is that for all the features putting together, their experimental measurements, their background measurements, they are mutually independent. So you see, this is the setting we're considering. Say the feature is a gene, we consider two features. So conditioning on their mean, the measurements just randomly scatter around the mean. And this is the case we're working on right now. But a common case in real data is like this. Okay, so the two features may have correlations in their expected values, like the three dots here. And their measurements, although being independent given the mean, but if we just look at the measurements before aligning the expectations, we will see the two features still have a correlation. So to address this problem, it's actually our next step. We are going to try to align the three means to be the same. And in biological analysis, it's usually called batch effect removal. So we want to make the data comparable. So this is the next step. But for now, we are working in this scenario. And also for a feature J, if being uninteresting, we are going to assume that their M and N measurements are identically distributed. Okay, so sorry.
So sorry, I have a question. So Jessica, can you go back to the previous slide? For your second assumption, is that also conditional on mean or it's unconditional?
It is conditional on mean, yes.
Okay, so even for uninteresting features, they can have different means.
They should have, actually, that's a very good question. Let me think. So they are identically, oh, no, actually, it should be, no, it's not conditional. I should say this. Yeah, so because we are assuming uninterested features share the same mean.
Okay, sure. Yeah. So then, just a minor suggestion. Maybe you want to put the conditioning on mean part inside of the independence. I mean, inside of the first condition.
Thank you. That's a very good suggestion. Yeah.
Thank you.
Okay. So now we're going to say how we design the contrast scores for three analysis. Enrichment with equal replicate number under two conditions, m equals n. This is our easy start. The reason is that in this case, we have the same sample size under two conditions. So the background observations naturally serve as the negative control or knockoff for the experimental measurements. So we don't have to do any construction. We can work with the data directly. So we start from here. Then we will talk about what if m doesn't equal to n in enrichment analysis and differential analysis. So step by step. So in this contrast by calculating the two means of a feature under two conditions and do a difference. Because the difference will give us the sign that will indicate whether this X bar is larger than Y bar. If the sign is positive, we would know, oh, this feature is likely to be interesting, right? Otherwise not. So this is and we also obtained this because this is a lot of biologists do, right, in their analysis. And the second summary statistic was motivated by the knockout paper. So by taking the larger one of X bar and Y bar here, I don't need absolute sign because they are non-negative, and multiply by the sign about the direction, right? Which one is bigger? So using those two, we just define contrast scores directly in this way, in the simple setting with n equal n. Okay, so this is a very simple illustration of m and n being 1. What do we do? So here for this feature, 10 and 0. For minus, it's very easy. 10 minus 0, 10. For the max, it's 10 times a positive sign, so 10. Uninteresting feature, minus is 1 minus 2, negative 1. The max is 2, bigger 1, times a negative sign, so negative 2. Okay, so this is how we use this BC procedure next. So with our contrast scores, what we look at is we look at the contrast scores absolute values that are non-zero in the set. So possible thresholds are going to be found in the set, okay? And then we look at this ratio. So this is a histogram that can help understanding. So let's say that this is a threshold T. We count how many are on the right of it. And we also look by symmetry, the negative T on the left-hand side, we look how many are on the left of it with some small modifications like +1. And also this is to avoid dividing 0 so that this ratio is under Q. And for all those possible T's, we find the minimum T. That is, we try to push this boundary to the left as much as possible. That can give us the most number of discoveries. So this threshold is called TBC. And because of the theory in this paper, it can guarantee the frequentist FDR control. So we basically sit on their shoulders and try to use this result. So you see, here the case is that once we found this T, then any features with contrast scores to the right of it will be called as discoveries. So people may wonder, how does this procedure work, or how is it powered when people do have high resolution valid T values? So this is a paper that compares BC procedure in that case with a BH procedure, and shows that the BC procedure is not really lose much power and can sometimes be more powerful. So that gives us some confidence for this. And moreover, with some discussion with biologists, they really want to have a Q value for each feature J, so that they don't have to define this Q beforehand. They are just a mouse. They don't have to define this Q beforehand. They will just give a Q value for each CJ. So to achieve that, you see, we just want to do this feature in a reverse way. That means for each CJ, we just look at what is the corresponding Q if we set a CJ, if it's positive, as the threshold. So that can give each feature a Q value. Okay, so this is the start.
Yeah, Jessica, can I ask a clarification question? So for the contrast scores, do you standardize them?
You mean divided by the standard error?
Right, yeah, because I think BC procedure uses the same threshold T here, right? So okay.
That's a very good question. So back here, of course, we can have like a T-test statistic, right, by dividing the standard error. And we did that comparison in our paper. But very interestingly, it's not as powerful as just the difference. And we think the reason is because looking at this figure, so this procedure will be more powerful if we have a heavier right tail. But with the standardization, actually the histogram will be more symmetric. So even though both can control the FDR without a problem, but using the T statistic, actually we have smaller power. And that's very unexpected.
Yeah, that's very interesting, because I was thinking here you have D different variables. I don't know how you call them, the D different ones. So if they have very different variances, then using the same threshold can actually achieve higher power. Yeah, that's indeed very interesting. Mhm.
It's very interesting indeed. And we think so we think our intuition tells us that here, because when we do the, find the threshold, we are looking at the side-by-side comparison between left and right. So even though some features may have bigger variance than other features, but our idea is that for those uninteresting features, they may have, like, some uninteresting features may have larger variance with a positive contrast score. Some may have negative contrast score, but because we have that sort of, still symmetry for those uninteresting features, even though some may have bigger variance, some may have smaller variance. So that will not affect the control here. Yeah. So,
Okay.
but I agree, this is very counterintuitive. And we don't found this result. Yeah.
So
Thank you.
So this is related. If I remember, one of the reasons why people like the DS, the negative binomial parametric model was that with this type of data, you often have a very heteroscedastic behavior across different variables.
Exactly.
And so I was wondering, this procedure, it doesn't really matter how you order the variables. Right? So you don't have to, you can use any, there are actually many ways you can order the variables. And so you could, in principle, use some parametric model to determine the ordering as long as it doesn't break the simple symmetry.
Actually, yeah. Well, actually, you will see that in my last slide. we actually did that. That's something we're actually working on. But for now, I have to clarify, I didn't say this technical detail, is that when we apply this method to that counts, right, non-negative counts from RNA-seq data, what we actually did is that we used log x plus 1. We did that transformation. So for each count, we first add a 1, and then we do log transformation. This is sort of to reduce, that skewness of negative binomial distribution, but still satisfy our non-negative requirement. And also, the motivation of doing that is also because for many biologists, when they look at the differential expressed genes, they are often interested in the quantity called log ratio. So you see the log ratio is very similar to what we work with the X and Y here. So that's why we did that transformation. And we did find that that's very important, that step. So for us to apply this, minus thing.
Yeah, sorry, just one more comment. I think to follow up with what Matteo just said. I think the FDR is under control with unstandardized test, BC statistic. That makes sense. I think I'm curious about the power part. Yeah. And I think it's quite interesting that you don't standardize the test statistic and the power can actually be higher. Because my understanding is, if the heterogeneity is really very high, then the null feature by chance, they have good chance to exceed the threshold, right? So that also means that you may tend to select a more conservative threshold. So yeah, that's why I was wondering. But I think for the FDR part, there is no surprise that this procedure always controls the FDR.
Yeah, exactly. Yeah, so I agree. For the power part, our understanding is still quite limited. Yeah, so I think there are a lot of things that can be done to really understand this part. Yeah, and I was want to show some more interesting, I would say some other interesting results later. Thank you for the comment. Okay, so this is just the theoretical conditions that are needed for guarantee the FDR control. So yeah, so basically using our notations, we have assigned positive 0 or positive 1, 0, negative 1 for each contrast score. So we need those, signs. These signs are mutually independent. So this is guaranteed by our first assumption about the independence from there. And the second is that for any uninteresting feature, its probability of taking a positive sign must be no greater than its probability of taking a negative sign. And finally, for the uninteresting features, their signs must be independent of their absolute value contrast scores. Okay, so we can see that when m equals n, with our assumptions, these three conditions hold. But when m doesn't equal n, we would have issues with number 2 and number 3. So we need some fix. And this is also aligned with the knockoff literature, right? We need to generate a knockoff to make a contrast. So, but in our case, it's so much easier because we only examine one feature at a time. So we don't need to care about the features joint distribution. So what we did is that we just do a simple permutation of the observations, which are the n columns, So by doing a random permutation denoted by sigma, we obtain this new matrix. And in this new matrix, we just pretend the first n columns are from the experimental condition. The last n columns are from the background condition. So this will give us permutation measurement. Okay, so with the permutation, we have defined our negative control. So let's say that we don't know how many permutations we need, so we just know we do H permutations. And this is the original data denoted by sigma 0, the identity permutation. So together, now we'll have H plus 1 data sets. One of them is original. So we need to define something about feature J, and we call this degree of interestingness, right? So this is using the minus statistic again from what we defined before. So we will going to define an interestingness for feature J in the original data and each of the permuted data. So with those H plus 1 values, we will sort them from the largest to the smallest. Okay, after the sorting, then we define the contrast score as follows. So for the minus contrast score, we share the same name. What we define is that the largest minus the second largest, if the largest comes from the original data. So we still stick with the philosophy that if it's from the original data, means that the original data does better than negative control, we give the contrast score a positive sign. Otherwise, we reverse the difference, so it's negative. And for the maximum contrast score, we always pick the largest degree of interestingness, but its sign is depending on whether the maximum is from the original data or not. So still, the signs are shared between the two contrast scores, but we define them differently. Okay, so this is a cartoon illustration again. So if this feature is interesting, this is the original data. Now we are considering we have 2 replicates in experimental condition, 1 replicate in the background condition. So by our degree of interestingness, it's the average here minus the average here. So it's actually 8 minus 0, 8. And we did 2 permutations, for example. So with the permuted data, this average is 5 minus 6, negative 1, 3 minus 10, negative 7. So from those 3 values, we will define contrast scores. For the minus, it's the largest minus the second largest, so 8 minus negative 1, 9. For the max, it's the largest times the sign. Here, the largest comes from original data, so the sign is positive, so we keep it as 8. So this is an interesting feature. For an uninteresting feature, I'll skip the explanation, but this is how we get it, minus contrast score, max contrast score. So from there, we will find the cutoff, right? So when the H number of permutations equal to 1, we could still use the BC procedure as we did. But when H is greater than 1, we use this GZ procedure so that we basically normalize the denominator by dividing H. H is the number of permutations so that the FDR can be controlled. So for this GZ procedure to work, which was from the multiple knockoff literature, so we need those assumptions, again, independence, mutual independence of the signs. And here for feature J, its sign being equal to positive 1 must be no greater than 1 over H plus 1. So this is actually from that uniform distribution of the H plus 1 degrees of interestingness, right? When we sort them, if the feature is uninteresting, then there's a chance of 1 over H plus 1 that is the original data's interestingness that got the maximum. And also we need the sign to be independent of the contrast score absolute values. Okay, so that's for enrichment analysis with different number of replicates. Similarly, we can do the same thing for differential analysis, except that when we define the degree of interestedness, we added the absolute value, because here the direction being positive or negative doesn't matter to us, only the magnitude matters. So we added absolute value. But the contrast score cutoff is also set by the GZ procedure. So as a preview, in the last slide, I will discuss the connection between this and the six-step plus procedure used in the knockoff literature. So there's something we could do there. Okay, so for now, I'm just going to quickly show some simulation results we did for those settings. We consider enrichment or differential analysis For the sample sizes, I'm showing these 4 settings here, 1 versus 1, 2 versus 1, 3 versus 3, and 10 versus 10. And for the distributions, we consider Gaussian, Poisson, or negative binomial. And for the background measurements, we consider homogeneous, which means the features are IID, or heterogeneous, they're not IID. And for the feature number, we consider 1,000 or 10,000. Okay, so for p-value based methods for comparison, we actually consider those 4 pairs. The p-value is calculated by the paired approach or pooled approach, thresholding by the BH procedure or Q-value procedure. That gave me those 4 methods. For the local FDR-based method from Ephron, we consider the empirical null, which is by assuming that the null is a Gaussian distribution, or by doing the observation swapping between the 2 conditions for calculating the FDR. But I skipped the technical detail for this part. I'm still including the comparison here. Okay, so first, interestingly, we are showing just the result, limited result for the Q value being 5%, the target FDR. So ideally, a method should have the FDR to the left of the dashed line, and the power, the higher, the better. So in this ideal case, with homogeneous background, then not surprisingly, the pooled approach using either BH or Q value has the best power. But we are the second best. But when the background is heterogeneous, then the pooled approach will fail very badly. But we still maintain a good power. So for the local FDR the local FDR approach, we found it very interestingly that even though even though it's not really designed for exact frequentist FDR control, but its FDR is very small and its power is not that bad. Okay, so even though it's lower than ours. And for the 3 versus 3, here I'm showing the Gaussian distribution without outliers, data are homogeneous, heterogeneous background, or with existence of outliers. So you see, without outliers, then this BH pair correct means that we are using the correct t-test to do it. So you see, they're doing very well, but we are also doing very well. But when the outlier is there, then the t-test suffers greatly from the existence of outliers. Well, we are very robust. And for this 2 versus 1 and 3 versus 3, now the results are based on Poisson and negative binomial. So one thing I want to point out is that even with this correct, okay, by saying that, especially here, negative binomial, even though we assume the data follow negative binomial distribution, but if the sample size is so small, like 3 versus 3, the estimation of the dispersion parameter, which is an additional parameter besides the mean, would actually cause some inflated FDR. So if you don't use the true dispersion, the estimate from the data, that estimation uncertainty will actually make the FDR control not well. So this is something we found very interesting in this analysis. So finally, for 10 versus 10, the reason we include this is because this is the case when sample size is large enough, so we could apply Wilcoxon test or permutation test, or we can do the parametric test with the correct model. Here is Poisson negative binomial. But still, in this case, we still didn't do very well, comparably well. So we think this is very encouraging. Okay, so finally, I want to quickly just show some omics data analysis. Here, we compare our method against those popular bioinformatics tools developed for each task. Like for the peak calling, we compare to Max and another one, Homer. So we can find that here, they do have some inflated FDR issue, right? The ideal thing is that actual FDR should be below this diagonal line, but they're not. And with our method, so what we did is that we just used our method as a downstream step of whatever statistic they calculate and define contrast score and then do the cutoff. We see that we can greatly reduce the FDR below the diagonal line. And our power is still quite good. So we achieve very good power. And for the mass spectrometry, this data, I want to say this is a real data benchmark. So we obtained the benchmark data set from our collaborator. So we are calculating this FBP because it's just one value, it's not the expectation. And we found that compared to a popular method, sequence, we can actually achieve better power under the targeted FDR control. And finally, for those sequencing, like single cell data differential express gene and the high-C data analysis, we also see that like some method like this one has very bad FDR and we can control it. And also we are doing very well. Like when the FDR is greater than 2%, we can actually beat all the methods in terms of power. And also this is very crazy. You see those two methods have FDR almost close to 100%, which means that they're not controlled at all, but we can do it. So just to quickly summarize the talk. So this is our paper, which was published last month, maybe October, sorry. That we actually received considerable attention in the field. As you can see, the number of accesses is quite good. And so I think a lot of bioinformaticians realize this importance of p-value issues. So to summarize, what the two messages we gave is that it can control, Clipper can control the FDR, and it outpowers generic FDR control method with some p-values or local FDR, and it can improve or fix popular bioinformatics tools. So to discuss, there is some future direction. So this is what we have achieved, right? We can do this without relying on valid or high resolution p-values. And I think this is very important because in practice, how to derive valid null distribution, either finite sample or asymptotic distribution, is really sometimes mission impossible for those users. So that's why I think getting rid of the need for this is very important. And also, we want to echo the importance of validating FDR control in practice, because in many of those popular tools, the FDR control is bluntly assumed but rarely validated. So people just use FDR as a mask but never check whether it works. And for the future work, one clear direction is to incorporate feature covariance for boosting the power. And another question which is open is that which contrast score is better? And what's the best number of permutations? How to set optimal H? So I have to see an interesting result from the results I showed is that we actually found H equals 1. Just doing one permutation gave us the best power. This is very counterintuitive because we know in permutation tests, we always know the more permutations, the better. But in our case, it's not really the case. So we think this might be related to the balance between the case and control. Since in our case, we have only one case, so having many more controls doesn't seem to give us good power. But I will also talk about that in next slide. And finally, we want to implement it in many bioinformatics tools to let people use it. So this is the connection between Clipper and the selective 6-step plus by Barbara and Candace. So in this general procedure, what it does is that assuming p-values are ordered in some way, independent of the p-values, then we can actually, given a pre-specified C between 0 and 1, what we can do is that from this target Q and the C, we can find the maximum K, given the order, the position K in the order, so that the discoveries will be rejected by both this K plus and also the C. So the order must be before K plus and the p-value must be under C. So our Clipper using BC and GC procedures actually correspond to this procedure in that when our contrast was positive, using BC procedure, it corresponds to p-value 1/2. When the contrast score is non-positive, it corresponds to p-value equal to 1. And here our C is said to be 1/2. Using GZ procedure, when positive contrast score, the PJ is 1/H plus 1. H is the number of permutations. When CJ is non-positive, PJ is equal to 1. And here we fix the C as 1/H plus 1. But after a more careful thought, we found that here the C's allowed range is actually fixed at 1/2 for BC. So there's not much we can do. But for GZ, it can actually vary between 1/H plus 1 to H/H plus 1. So therefore, we may gain some power by changing the C, not restricting to this, but allow this to be optimizing this interval. So in that case, we may have a better power for GZ procedure. But I have to say that our current result is based on the C, so that we found actually H equals 1 gave us the best power. So we will leave this as our concern. Yeah.
So I think it's known that it's not clear how to choose C in selective six plus
Yeah.
when you have more than one permutation.
Yeah.
And I can see why having more permutations would be a problem with the GC procedure, but if you fix C, there really is no reason why you should use that C. In my experience, what works well with this kind of order p-values is Lee and Barber's accumulation test. Have you, do you know what it is?
No.
It's a different type of FTR controlling procedure that takes as input low resolution p-values with an independently given order. The difference is that it kind of has a smoother, a smoother, instead of having a constancy, it kind of like combines the p-values in a much smoother way. Basically, it doesn't really, it does something very similar. It has similar properties, but it doesn't have this parameter that you have to fix in a base.
So you don't have to fix C.
Basically, it's yes. And at the risk of oversimplifying, it's like this without fixing C.
I see. Thank you.
And it has all the same properties as similar martingale proofs of FDR control.
I see. Wow, this is very helpful. Yeah, so this is the reason why presenting work is very useful. Thank you. So finally, this is back to Mateo's earlier comment. We did try to add Clipper, use it as a downstream step of those popular tools like the DEC2. So we found that, you know, in this is a real data actually, but we're checking the actual FDR by using permutations. And you can see that using DEC2 and HR alone, they do have severe FDR inflation issue, but we actually using Clipper based on either their statistic, like the test statistic used, but not their null distribution, or we just use the log flow change output by those two methods. All four versions can control the FDR quite well. And among them, using Clipper plus the DEC2 statistic gave us very, very good power. So, you know, the original method, these two have perfect power, but using our version with the FDR reduction, we still achieve very good power. So although I have to say this is very preliminary, but it at least give us some hope that we can actually do this. So try to get rid of their null distribution, which might be the issue of causing the inflated FDR, but we can use the power in their statistics. Okay, so that's all. So I want to thank my two students, Shindra and Eileen, for doing a lot of those numerical work in this work. It's really intensive. And also the collaborators who give us the biological data set for the feedback. So, and the funding and uses as well. Thank you. Thank you.
Can I ask another clarification question? So for the permutation method to generate knockoffs, I wonder if you can clarify how that was implemented.
Okay, so in our current implementation, I should go back here. We just permuted the columns for once. So for each permutation, in other words, every feature shares the same permutation. But this could be done differently. Like we could permute each feature independently of each other.
And you put the two samples together to permute, not like within each sample.
Yeah, we put
Okay.
the samples together.
Yeah.
The purpose is to shuffle the labels so the two conditions are not really two conditions.
Sure. Okay. Yeah. Then I'm curious because you mentioned that when H is equal to 1, the power seems to be the best. Is that possible that when you permute and then you do a contrast, you sort of reduce the signal strength because there is an averaging process. Like then the more you permute, it feels like then you are averaging across a larger number of like permutations. So if each one was reduced the signal strength, then maybe that will reduce the signal and reduce the power.
And that makes a lot of sense because here we are doing this ordering. So like you see here, the larger the age, actually we can see the more likely that an interesting feature may not get the maximum. It may go somewhere in the next, like in the middle here. So in that case, we will actually receive the negative sign. Yeah, I think that is a very good point. I think it was from a epidemiologist who said that they know in the case control study, balance would be better, not really having more control would help.
Right, yeah. Well, probably if there's another way to do, to generate a control, then the power can be preserved. But for this particular permutation method, I suspect that that will reduce the power because of this averaging process.
That makes sense. And also another thing we think is the reason I said we could do this permutation for features independently, although that would definitely be more computational time. But why doing that might be better is because if we permute the data independently, at least when we define this degree of interestingness, let me see here. We would define the degree of interestingness, at least for these things, right? We can make them independent for features, even though we may not satisfy the independence assumption for D features on the original data. But at least for the control part, we can make them independent.
Mm-hmm.
Yeah.
Okay. Thank you.
Thank you.
Maybe just a question about the interpretation of the hypothesis, because I thought you started at the beginning by saying that we wanted to compare the means in the two groups, but now we're actually testing whether the distributions are the same, right? Is that something that is the difference relevant between those two assumptions?
Actually,
or
I have to say that is actually a very good question. This is actually part of the contrast score design. So right now we are mostly using this, right? So the average differences. So to us, this only captures the mean difference, because this is like an estimator of the mean difference. But if we want to go to, but I'll agree. So for the assumption part, when we assume the uninteresting features, we are actually assuming additionally that they are IID, not just mean equal. So I agree, that's something that, but I see that more like an assumption, and it's gap from the setup. So but back to your question, if we want to do the distribution difference, not just the mean difference, so one option to consider is to replace this statistic by, say, the Wilcoxon statistic, which basically is a non-parametric way of doing this difference in distribution instead of just looking at the mean.
I guess what I was wondering about is whether there is some situation where the distribution in the two samples is not the same, but we're not interested in it unless it's a difference of means.
Okay, the distributions are not the same, but we're not interested unless it's the same.
Or is that not?
I would say theoretically, yes, but I don't see much in practice. Yeah, I think the reason is that seeing mean difference is more easy or more
I
intuitive.
see. So people were testing means just because it's easier, not because they actually care about the means, especially.
I guess so. Yeah, because I know in this bioinformatics field, I do see some statistical based people. They propose many different tests for testing the variance, for testing the modes, whether there are two modes versus one mode. So there are methods like those, but unfortunately, they're not commonly used. Yeah. So I would say the major reason was people's understanding is this. Especially a lot of people, they just look at the two conditions as two box plots and see visually whether one is higher.
Well, then, we're perfectly out of time. Thanks a lot.
Thank you,
Well, I have more questions, and I'm sure others have, but we have all day to ask you. So
Okay,
thanks a lot.
great. Thank you.
Thank you.
Thank you.
Yeah, I'm sorry. Yeah, I'm sorry. It's a small. Yeah. I'll see you on the. Or I guess we're going
But I joined the.
Yes, yes. that's fine. So where's in the rest of the room? Are you going to?
Yeah, so I think I'm just going to the office. That is in the fourth floor. Yeah, yes. I have it.
Okay, let me just sign.
Okay, thank you. Yeah.
Thank